Hello everyone. Today we will study about enzymes. As in previous lecture, we have studied about what enzyme is and what is the mechanism of the enzyme action. Now in this lecture, we will study about the different factors that affect enzymes activity. Enzyme activity means the measure of the rate at which an enzyme convert substrate to product in a biochemical reaction. Four factors that affect enzyme activities are temperature, pH scale, substrate concentration, and enzyme concentration. Now we will start with the temperature. What happened with the increase in temperature? With increase in temperature, we can see that enzymes activity enhance with the increase in temperature initially, but after which further increase in temperature, rate of the reaction shows a decline. From the graph, we can see that initially with the increase of temperature, the rate of reaction increases, but reaching at an optimum temperature, after that the rate of reaction start decreases because with increase in temperature, the enzyme stops its activity. Next one is pH. What is the effect of pH on enzymes action? pH, or we can say hydrogen ion concentration. Again, in this, each enzyme has an optimum pH value which shows maximum activity. Below this pH value or above this pH value, enzyme activity become low. As we can see, the enzyme activity increases at the optimum pH value and decreases after that. We can define pH from 0 to 14, like from 0 to 7 it is acidic, while 7 to 14 it is basic in nature. Next one is substrate concentration. What happened when we increase the concentration of the substrate? If the concentration of the substrate, or we can say reactant, is increased, the velocity of the reaction increases proportionally in the beginning, but it reaches to maximum value and thereafter no further increase in the velocity is observed. We can see this from the graph that initially with the increase in substrate, rate of the reaction increases, but reaching at the maximum reaction rate it becomes saturate. Now enzyme concentration. The rate of reaction increase linearly with an increase in the concentration of enzyme. That means if the substrate is available in excess, by increasing the enzymes concentration, the reaction rate will be increased linearly. We can see this in the graph that how enzyme concentration and reaction rate related to each other. Enzymes are not consumed in the reaction and enzymes can be reused many times. The next one is enzyme inhibition. What is enzyme inhibitors? A substance that slow down or stop the normal catalytic function of an enzyme by binding to the enzyme is called enzyme inhibitor. Basically, there are three types of enzyme inhibitors: reversible competitive inhibition, reversible non-competitive inhibition, and irreversible inhibition. What is reversible competitive inhibition? Competitive inhibitors are those inhibitors that compete with the substrate for binding to the active site of the enzyme. If an inhibitor is bound to the active site, it prevents the substrate molecule to access the active site, hence decrease the enzyme activity. We can see from the figure how enzymes and the competitive inhibitors compete each others, and if competitive inhibitors bind to the active sites, it slow down the reaction rate. Next one is Reversible non-competitive inhibition. Non-competitive inhibitors decrease enzyme activity by binding to a site on the enzymes other than an active sites. By doing this, it decreases the enzyme activity and substrate cannot fit into the active site. Process can be reversed only by lowering the non-competitive inhibitors. We can see how this activity happened from the following figures. And the last one is irreversible inhibitors. And irreversible inhibitors inactivate an enzymes by binding to its active site by a strong covalent bond. Means it permanently deactivate the enzymes. Irreversible inhibitors do not resemble substrate, chemical warfare and insecticides are the best example of irreversible inhibitors. Here is the table given below through which we can summarize what is enzyme inhibitors and its type. Like we already discussed, it is the substance that bind to enzyme that stop or slow the normal catalytic activity, and types are competitive enzyme inhibitors, non-competitive and irreversible enzyme inhibitors. How these inhibit the enzyme action is shown by the given figure. Thank you.